Hello and welcome everyone. So in the last class, we learned about how we can use MNA sequencing or MNA's qPCR to get information about nucleosome positioning. So today we'll learn about the how we can alter the access of restriction enzyme on a linear array of chromatin and how it will give us the information about the nucleosome positioning. So uh, this is linear array model of chromatin suggests that chromatin is kind of a beads on string structure. Okay, so this beads on string structure is capable of nucleosome sliding as well as nucleosome ejection, eviction, all those things. So this can be done with the help of chromatin remodelers. We have already learned about this in the previous video lectures. So, uh, so this is the linear model. This is the bits on the string chromatin structure. So let's assume that this string has two sites for a restriction enzyme. Let's take EcoR1 for example. So this EcoR1 has two sites, this restriction enzyme has two sites. This is site A and this is site B. Okay, so here you can see site A is on the nucleosomal DNA. It is occluded by nucleosome, but the site B is on the linker DNA. Okay, so what happens that when this nucleosome slides further, you can see that site A is getting accessible. Okay, so this goes in the linker DNA, so it is getting accessible, while the site B is getting somewhat inaccessible because it is very close to nucleosome, so it is inaccessible. So due to the dynamic nature of the nucleosomes, the sites can get from accessible to inaccessible or vice versa. Okay, so how can we know about the nucleosomal positioning by using restriction enzyme? So these restriction sites help us. So you can first you have to simply isolate the chromatin from the nucleus, and then after isolating chromatin you will digest it with the restriction enzyme, which has different sites on the chromatin, multiple sites on the chromatin. Okay, and after the digestion you can either perform gel electrophoresis or sequencing or qPCR, quantitative PCR, for for collecting the data, and this data will now tell you about the accessibility or inaccessibility of the sites. So if the site is accessible, if site is accessible, then it will be cut. Okay, and the if site is cut, when is site accessible? Site is accessible when it is not in the nucleosomal DNA. So when it is not in the nucleosomal DNA. So let's assume that it is in the linker DNA, and when site is inaccessible, okay, so if site is inaccessible, of course, the restriction enzyme won't be able to cut it. So it will remain uncut, means it is the nucleosomal DNA. Okay. So scientists collected the data and they plotted the percentage cut of the cut on the sites A and sites B over the time. Okay. So let's assume in the case one, this is the case one and this is the case two. So in the case one, what happens that the percentage cut over the time for site A, it's somewhat this. Okay, so this is profile for A. So this is the profile of the site A, percentage cut of site A over the time. But when they observe the same for time B, the percentage cut over the time for B was much higher. So this is the profile of B. So this tells us that the B, due to the percentage cut over time, means the slope is higher. It states us that site B here is accessible. It is more accessible than site A. Okay, so this tells us about the case A. But when they performed the same procedure, the same experiment for the case B, when after the recruitment of chromatin remodelers, so what they observed was they could when they observed they collected data and plotted it percentage cut over the time. The profile for B was this, while profile for A was somewhat like this. So this time they got to know that A is more accessible than B. Okay, so this is how scientists, the researchers were able to tell that nucleosomes are the dynamic structure which can undergo sliding, eviction, assembly, disassembly, and all with the help of, or due to, or you can say due to, chromatin remodelers. Okay, so this thing is a well established fact, and now using the restriction enzyme, which has multiple cut sites or overall on the chromatin, you can predict the nucleosomal positioning. Okay, so you can exactly predict that the site when the different sites for the same restriction enzyme are either occluded by nucleosomes or not. So that's it for today. In the next video, we'll learn about some other assays. Till then, see you. Bye bye.